were talking last time about, we ended, I think, talking about how DNA is a language and how you could code German or you could code English or you could even code Chinese into nucleotide language. And I was just perusing some of the science blogs that I read, and just this month in Science, these two gentlemen, this is George Church and his postdoc Sriram. They just published a paper actually this month talking about how they've been encoding things into nucleotide language. And their breakthrough, now people have been doing this for a while, and we'll talk about synthetic genomes and people doing DNA watermarks, but what they're really trying to do is press the limits of how much information can they actually pack into DNA. So they took George Church's recent book, and they took the entire book with illustrations and everything, and they coded it into binary. It's available online in HTML format. You can go online and buy his book, read his book. And I mean, HTML is just written in binary code, right? Zeros and ones. So what they did is they took that entire HTML version of the book and they coded it into DNA. And they came up with a density of 700 terabytes per gram of DNA. 700 terabytes. Think about how much that is. I mean, they make some big hard drives now, but I think 3 terabytes is about the biggest one you can buy, right? So 3 terabytes hard drive, you would need what, like 230 or so? 230 3 terabyte hard drives. That is close to, it's over 300 pounds. Okay? That is the densest technology we have for coding information. 700 terabytes, over 300 pounds. You can get all of that information into DNA in 1 gram, right? You can dissolve that in 1 milliliter. 1,000 microliters, you could dissolve 1 gram of DNA, and you could have 700 terabytes of information there. What they do is, can you see this? It's kind of hard to see, but this is what they did. Here's a sentence out of his book. So, decoding self-referential DNA encodes these notes. They took just this blue part, okay? And they're showing you an example of how they encoded it into nucleotide language, okay? So this is the binary for that. Referential DN. That's encoded in here. So 0110011. This is the ACSI, ACS2. Binary coding for English, right? So 011001110 codes for F, right? So they've encoded each one of those letters into binary code. That's the HTML code for this. Then they encoded that into DNA language. So for an F, they encode it. Basically, they're just using binary, right? And they're just using G's and, I think it's, what is it, G's and C's for 0 and A's and T's for 1. So it's a pretty easy conversion, right? You just, if you want a 0, you put a G or a C. If you want a 1, you put an A or a T. And they just encode it into nucleotide language. They synthesize these artificially. So we have machines now. You just throw in a bunch of DNTPs, deoxynucleotide triphosphates. You put them into a machine, it does the organic chemistry for you, and it synthesizes these pieces. So they synthesized a bunch of tiny fragments that encoded these little fragments of sentences in his book. And they also did this for images in his book. If you look at an image on the screen, we're looking at the picture here. That picture on my hard drive is coded in 1s and 0s. So you can code for pictures just by taking the binary code 1s and 0s that codes for that picture, translating them into nucleotide language, and putting it into synthesized pieces of DNA. So they synthesized a bunch of these pieces. They all have a little tag code on them. That's what's in red. It's a little organizational feature so that you have all these fragments. Those tell you how to put the fragments together in the proper order to read the book. So you can actually read his book by sequencing all of these pieces of DNA, organizing them together using a computer to do it, and read the HTML based on DNA language. You could do this for video as well, right? You could take video, digital video, convert it into nucleotide language, save it as a piece of DNA. This means that this makes DNA the densest form of information storage that we've ever found. Densest storage material known. First of all, it's volumetric. All of the storage things that we use are disks, like a Blu-ray disc or a hard drive disc, right? It's a linear, flat, one-dimensional thing that we're encoding on. Well, DNA in solution or DNA as a powder, I mean, it's, you can compact it into a volumetric thing, so you can get that really dense. You don't have to keep moving out in a linear form. You can keep packing it into a dense volume. DNA is actually really stable over time, and DNA is redundant here as well. So if I'm coding something on a disk, I'm typically just only coding it in one spot, or I'd have to code it in 2 spots and take up more space, right? So then I quickly get a lot of space saved up. Or, my space is expanding. And then hard disks, Blu-ray disks, are only going to have a certain lifespan. They're only going to last as long as the physical plastic is going to last. And a hard drive is even more technical and prone to error, right? You got to keep them cold. You got to keep them running. There are physical things in there that could break. If you break the hard drive, then you've lost the data, right? DNA is actually very, very stable over time. If you took all these pieces of DNA, dehydrated them, and put them in a bag, they'd be stable at room temperature for hundreds of years. Much more stable, actually, than the plastic or the metal or whatever you're writing things on a disk is. Room temperature, so you don't have to worry. Like a lot of Google's storage centers, or Amazon's storage centers, or Netflix storage centers, all these people that are compiling all this DNA, or all this information and storing it, they have big facilities, usually in really cold places. Like there's a big one in Norway and in Sweden and in Canada, and they have these big warehouses so they don't have to pay so much cooling costs to keep all that storage cold. DNA, you could just be storing it at room temperature and it wouldn't decay. And also, it's redundant. So if the DNA does decay, I could have dozens of the exact same information, and since I can pack it so densely, you can afford to be redundant. So then if I had a mutation, a decay, a damage to one of the things, you could have damage, and that's what these little dots are. You could have a damage at various spots, but if I read a dozen of them, I could figure out which ones were damaged and which ones were not. So the redundancy keeps it even higher fidelity. So there's a lot of people that are actually working on trying to adapt DNA and use biological storage. Since it's so dense as well, think about that same 300 pounds of hard drive space, right? To code in 700 terabytes in our hard disks, that's going to be 300 pounds. If I can do that in 1 gram of DNA, think about how much information you could store if you had 300 pounds of DNA in a warehouse. You're closing in on all the information content that we have ever produced in the human race, right? We could have been videoing everything since the dawn of time and putting it into digital and probably get it into that 300 pounds of DNA. It's pretty amazing technology. This was done at Harvard's Weiss Institute. And I just ran across this yesterday, and I watched this little video. George Church was talking about doing this. And at the end of the Weiss video, it gives their mission statement. It says, the Weiss Institute uses nature's design principles to develop bio-inspired technologies that will revolutionize medicine and create a more sustainable world. All they've done here is substitute God and put nature in there. My thesis is nature, chemical laws, physical laws don't produce anything. They don't design anything. Persons design things. And this just makes me think of Romans 1. For since the creation of the world, God's invisible qualities, his eternal power and his divine nature have been clearly seen, being understood from what has been made, so that people are without excuse. For although they knew God or knew about him, they neither glorified him as God nor gave thanks to him, but their thinking became futile and their foolish hearts were darkened. I love these examples of reverse bioengineering and finding if God has made a material, hundreds of times more effective in coding information than we have ever created. So I think that's a powerful motivation, I think, for you guys to be involved in science, and I think also a powerful testimony that we can give to people who are involved in science, that these things don't happen by chance, that information is coded, it's designed by somebody, and we get to know who that person is. Last time we left off talking about DNA. We talked about double-stranded DNA. We talked about hydrogen bonding, holding the double helix together, and we ran out of time, and so we need to start talking about RNA. Like I said before, RNA differs from DNA in only two ways. Instead of a thymine base, there is a uracil base, but the code is still the same, right? Just T and U are substituted. There's no real change in the language. The other is that the third carbon still has its oxygen on it, whereas in DNA, that third carbon doesn't have the oxygen. There's just a hydrogen. Other than that, chemically, they're identical. When you isolate nucleic acid chemically, you're going to get DNA and RNA. You're going to have to do something to get rid of one or the other if you want to get one in isolation. So chemically, they're going to act very, very similar. Structurally, though, they're different, because most of the time, RNA is made in single strands, and there isn't a complementary strand on it. Now, there are some exceptions to the rule. There's some viruses that have RNA as their genome, and they'll have double-stranded RNA, and they use that. But most organisms use DNA as their genome and have it double-stranded. DNA and RNA, though, really dislike being single-stranded in terms of just they don't flop around like a floppy noodle and have all of their bases just exposed. They really want a hydrogen bond. The bases are going to want a hydrogen bond to each other. So even though RNA is mostly single-stranded, it's going to take conformations like this, where it folds back onto itself. Okay? So here's the 5' end of this single-stranded RNA, and here's the 3' end. And RNA forms what are called stem loops. So here's a little stem where this 5' end wraps around itself, and it forms a stem where hydrogen bonding occurs. That hydrogen bonding is the same type of hydrogen bonding that's happening in DNA. G's and C's are pairing up, making 3 hydrogen bonds. A's and T's are pairing up, making 2 hydrogen bonds. And so we're doing canonical base pairing here in the stem. Now then there are going to be regions that don't have any reverse complementation. So those are going to exist as loops. And RNA can actually have some very elaborate and really cool secondary structure like this. It can fold on itself in some really interesting and actually enzymatically active ways. So RNA will actually sometimes be able to perform chemistry because it's in a certain conformation and can actually catalyze chemical reactions. Most of that's due to its secondary structure. So here's lots of stem loops here. There's going to be some spots that don't make any secondary base pairing, but you're going to have lots of these little loops and stems going on. This is going to be the case for messenger RNA. So when the cell makes a copy of the DNA that it wants to express as protein, it's going to make an RNA copy, messenger RNA, and it's going to fold on itself like this. Transfer RNA, tRNAs. These are the ones that are actually involved in converting a messenger RNA sequence into a protein sequence. And these are a very specific secondary structure as they fold on themselves that make them capable of moving those amino acids around. And ribosomal RNA. Ribosomal RNA is huge. These big, long RNA strands, there's multiple ones in the ribosome, and they fold in really interesting ways and make the whole translation process possible. So we'll go more into RNA, but I just want to give you an idea. DNA just never wants to be a floppy noodle. It's always going to want to stick back on itself. So that when we deal with RNA in the lab, we're going to have to deal with this as well. We're going to have to manipulate it. You usually have to boil the sample of RNA to get those hydrogen bonds to break so you can do something with it. Whereas DNA, when it's double-stranded, at least it's not folded in weird conformations. Lastly, there's different helical structures that DNA can have. So this will end our structural talk, and then we're going to shift into genomes, talk about larger pieces of DNA. But DNA can exist in 3 separate forms, at least 3 separate forms. I think there's more, but these are the big ones. bDNA is what you typically think about when you're thinking about the DNA double helix. This bDNA is DNA in aqueous solution, in water-based solutions. And water is going to be nestled all in between the base pairs. It's going to be nestled in between the 2 strands. And it's going to hold DNA in this conformation. This is a right-handed helix. So as the molecule turns and spirals around itself, it turns in a right-handed manner. And there's certain standard gaps between the minor groove and the major groove that bDNA takes. And this is mainly just properties of it being dissolved in water. Okay. aDNA, you can see aDNA, it's still a right-handed helix, but it's kind of been compacted. The major groove is much smaller than the major groove in bDNA. It's a little bit closer to being a symmetrical helix. This usually happens when DNA is dehydrated. So we're going to precipitate DNA out of solution by adding a lot of ethanol. If you put in a lot of ethanol, you displace all those water molecules, and the DNA molecule kind of scrunches down, tightens up its helix. It's a little bit denser there. These are the 2 structures that we will have to worry about in lab, and in most things you're going to do. You're going to have bDNA in solution, aDNA when it's precipitated or when it's dry. zDNA is possible to make. I don't even know what you have to do to make zDNA. I think it's in some organic solvents or something that gets it to turn into zDNA. It actually switches and is a left-handed helix now. So instead of spinning to the right, the helix spins to the left. And yeah, I'm not even going to talk about it too much because I don't even know if it even exists. There's some evidence that maybe some weird bacteria or cyanobacteria or something actually might keep zDNA in this form. But for anything that you're going to work with, it's probably not biologically relevant. We don't even know if there's some evidence it might exist in nature. We have no idea what it's doing. So I mean, it's kind of interesting, but I don't think it's terribly relevant. So just know that it can. Know that it's a left-handed helix instead of a right. But all right, questions about structure here. We're gonna switch and start talking about genomes as a whole. So are there questions at this point about DNA sequence, DNA structure, RNA sequence, RNA structure? Yeah. It's a left-handed helix, yeah. That's all you need to know. I will take no more questions on zDNA. We're good? Okay. Genomes. We're going to have to go through some definitions here for a minute, so it's a rather boring slide. Genome is the full DNA sequence of an organism's haploid set of chromosomes. So probably within that definition, there's already some further definitions that we have to work through. But a genome is the haploid set of chromosomes, the sequence of the haploid set. So what is ploidy? Haploid? The term ploidy means what is the actual number of sets of chromosomes that an organism has. So the ploidy level is, if you're a monoploid, that means you have one of each of the chromosomes. Bacteria and simple organisms will have only one chromosome. You have, how many chromosomes do you have? Depends on if you're male or female, but what's that? 46, yeah. You've got 46 chromosomes total. You have 22 unique chromosomes. And then if you're a female, you've got two additional ones. You've got one additional one. You've got an X. You've got two copies of it. So you have 23. If you're a male, you've got 23 plus a Y, right? Ooh, genetic diversity. The Y does very little. No, I take that back. The Y is very small. So we are diploid organisms, right? So we have 23 different chromosomes, and you have two copies of each of them. So 23 times 2 makes 46 total chromosomes. But your ploidy level is diploid, two-ploid. You've got two copies of every chromosome, okay? So organisms that have two copies are diploids, and like in the X and the Y, technically males are diploid, but I've got an X and a Y. So the chromosomes in the ploidy level could be slightly or significantly different in terms of the X and the Y. A tetraploid organism then would have 4 copies of every chromosome. Salamanders, frogs, some reptiles are tetraploid, have 4 copies of each chromosome. Some fish, zebrafish is notoriously hard to work with because and some frog species, they're like tetraploid or pseudo-tetraploid when they sometimes have 4 copies, other times it's not a complete 4th copy of each chromosome. That makes them really hard to do genetics with, so most genetics is done with a diploid organism or a monoploid organism. Alloploid. This is an organism that has chromosomes from different species, different sources. A good example of this is wheat. Wheat actually is, I didn't write it down. Wheat it's got 6 copies of each chromosome. So sextuploid, I guess. 6 copies of each chromosome. But actually, they're 2 sets from 3 different species of grass. So there was a forced hybridization between 2 species of grass. So both of them were diploids. So you force a hybridization between the 2 of them. Now I've got a plant type that's got 4 copies of the chromosome. And then you get a 3rd type of grass and fuse it with that one. And then you have 6 copies of each chromosome, all of them coming from an original species of grass. So wheat is a very hybrid organism. Plants can do this. Plants are weird. You can force them into multiple ploidy levels. So like most tomatoes that we eat, like the original ancestral tomato was probably a tiny little berry, probably smaller than even a cherry tomato, very small. But through selective breeding and forcing, sometimes you just naturally get additional chromosomes, like it goes through meiosis, or it goes through mitosis, you copy all the chromosomes, but then the cell didn't divide. And so I've got a cell that's got 4 copies. And sometimes that actually acts as a gamete, and you can get a forced higher ploidy level. That usually means that the organism gets bigger. So most of the tomato species that we eat now are tetraploid, but probably the ancestral tomato was only diploid. And so now we get these bigger fruits, and we've kept breeding them for bigger and bigger fruits. There are some species that can interbreed. So a lion and a tiger can interbreed, and you can actually get ligers or tions. And that's a forced fertilization between both of them. So that would be a lion or a liger or a tion is going to be an alloploid organism, right? They've got multiple chromosomes from different species. I think it's named after the male, so male comes first. So a liger is a male lion that mated with a female tiger, and then a tyon is the reverse. There's weird animals that you can do this with. You can get some whales and dolphins, so you get wolphins, or mules, right? Donkey. Yeah, so a donkey is a, what is it? Is it a donkey that's a mule and a horse? Is that right? Yeah. Most of those organisms are sterile because they have real problems separating the chromosomes. The cell doesn't know how to line them up. So they can copy all of them and separate all of them, so you can go through mitosis, and so an embryo that's a hybrid can grow into an adult form. But when it goes to make gametes, it kind of doesn't know how to line up the lion and the tiger chromosomes and separate them. So I think some donkeys are partially breedable, not very good. But ligers and tyons, you can't keep them going. And if you've ever seen pictures of a lion, or a liger or a tyon, they're actually really big, yeah. They're like a third or more bigger than the actual one. They're really bizarre. So I should have got pictures for you. Maybe we'll do pictures next lecture. All right, we'll start talking about really small genomes, and we'll build up to bigger ones. And I'm just going to give you some facts about the relative size of genomes. So here's some examples of viruses. Here's a viral genome in the bacteriophage. This is a virus that specifically infects bacteria. It's got a double-stranded DNA chromosome, but it is a single, only one chromosome, so it's monoploid, and it's a linear chromosome. So it's pretty small. Now, are viruses alive or are they not? I don't know. Because they have to have a bacteria to replicate, so they're not self-replicatable. But they have a unique genome. It's a really stripped-down genome because they don't have to do any cellular processes, right? All they got to do is make a couple of enzymes to hijack the bacteria to make more viruses, and then they get out. I don't know if that's alive or not. Retroviruses. A lot of retroviruses have RNA genomes, some of them single-stranded, some of them double-stranded. So they actually inject their RNA genome into a cell and then copy that back into DNA and then use the DNA to make more viral proteins and viral genomes. So I forget which retrovirus I'm actually showing here. This one was single-stranded, and it's actually got 2 chromosomes. So you can have a monoploid or a diploid RNA genome. The viral and the prokaryotic world is strange and bizarre. So you can have DNA chromosomes, you can have RNA chromosomes, you can have double-stranded or single-stranded, you can have linear or you can have circular. There's all kinds of varieties of how these chromosomes are, what they're made up of and how they're arranged. Yeah?
For like the viruses like the flu virus, that are segmented, what?
You mean segmented and they've got multiple chromosomes, right?
Yeah.
Yeah.
They would have.
Yeah, I'm not sure about the flu. I don't actually know much about the flu genome, the influenza virus. But yeah, it's got multiple chromosomes. I think it can vary in the number of chromosomes it has, but yeah. Prokaryotic genomes. There's kind of the most variety in the viral genomes. In the prokaryotic genomes, that's single-celled, non-nuclear organisms. Most of them are monoploid. They've got a single genome in one chromosome. Most of the time, in fact, I don't know of another example. I think they're all double-stranded DNA genomes. And most of the time, they're circular. So they don't have any 5' or 3' exposed ends. The 5' and 3' on one end and the 5' and 3' on the other are just hooked together. And so it's just a continuous circle of DNA. They're usually clumped together. So that circular genome will be clumped together, and it's kind of usually partitioned on the side of the membrane, on the inside of the cell. So they don't have a true nucleus. There's no membrane around the genomic and the DNA material. But they're often clumped together, partitioned to the side, and tethered usually to the inside of the plasma membrane. And we call that dense clump a nucleoid. So it's not a true nucleus, but you actually can look at a bacteria and see the DNA content kind of sequestered to one side. That's the genome. Prokaryotics and most bacteria in general usually have accessory chromosomes. They're not considered part of the genome because they often contain genes that are really not essential for the bacteria to live, but it's a means by which bacteria can exchange information with each other. So they'll sometimes have duplicate genes on there or unique genes on there that's not essential necessarily for it to do metabolism, but might give it some additional property, and they can exchange these. These are called plasmids, and they are also circular chromosomes, but they're usually very, very small in comparison to the genome. We'll talk about bacterial genetics and how they swap that information and then how we can actually manipulate it. But I just want you to know for now, that's not technically their genome, it's an accessory plasmid or an accessory chromosome. When we talk about the size of a genome, we're just going to use abbreviations for how big it is. So we typically talk about genomes in terms of their megabase or the kilobase size. So 1 kilobase is 1,000 base pairs. So I'm going to abbreviate base pair as just lowercase bp. And then you typically give it a letter to indicate how big it is. So a kilobase is a kb. Actually, that probably should be capitalized. A megabase is going to be 1 million base pairs. And here's just kind of the range and the size that chromosomes can come in. The human genome is 3.4 billion base pairs. That's actually gigabases. It's actually not the biggest. You can imagine wheat that's tetraploid, and there's 3 different genomes basically in there. Is much bigger. Corn's much bigger. All these plants that can be forced into greater and greater hybridizations will have really enormous genomes. The fly genome, 180 megabases. The E. coli genome is only 4.6 megabases. The smallest genome is in a bacteria called, I guess the smallest genome in a living cellular organism is a tiny little organism called Mycoplasma genitalium. Mycoplasma, it's got a very small genome, so it's a small bacteria with a small genome. And genitalium, it actually lives on usually the genitals of mammals. So it's a tiny little bacteria that kind of just lives in the genital tract, or there's lots of bacteria all over your body, actually. So that's the smallest genome. I actually forget the size of it, but it's smaller than E. coli genome, so I'll look that up. Plasmids are even smaller. They may be 1,000 or maybe 200,000 kilobases, so pretty small little guys. They can get them really tiny. The human genome, 3.4 billion base pairs. It took us several years to sequence through that using traditional sequencing methods. We have sequencing methods, and we'll talk about these later, how they work, but we've got sequencing methods that will do 3 billion base pairs in a couple of hours now. We can sequence really, really quick, so there's loads of organisms that have their genome sequenced. Usually they're just freely available. You can just go peruse them if you'd like. Most of this, well, we talked about Craig Venter before. He's the one that did the shotgun sequencing. He was competing with the Human Genome Project to sequence the entire human genome. That was his company Celera that kind of pioneered this shotgun sequencing. That was actually traditional sequencing. He was just fragmenting it and then just sequencing them all in the traditional method. We've developed even better ones that are like using fluid dynamics and tiny little tiny little droplets of water arrayed on tiny little chips doing multiple sequencing reactions all in parallel. We'll talk about that, but I want to talk about Venter because he's done something interesting, and actually just recently he's done something pretty cool with genomes. So first he had Celera Genomics, that was the human genome competition. Then he opened a company called the Institute for Genome Research, Tiger Labs, and this was a for-profit company, and they were just sequencing genomes for people. So if somebody wanted their genome sequenced, they'd get a grant from the government, and they'd pay the Tiger Labs to sequence it for them. He also started the J. Craig Venter Foundation, And this was one of the projects that this foundation did is Venter is really into sailing. So he got on his sailboat and he just wanted to sail around the world. So he was going into the open ocean, and he would just periodically put down a collecting bucket or something, and he'd collect water samples at various different depths as he was sailing along. And he would just send that water sample back to his labs at the Venter Foundation. They would just extract any DNA from any bacteria or algae or anything, diatoms, whatever was growing in that water sample. They would just extract the DNA from whatever was there, and they would just sequence it. And they were coming up with very unique sequences that no one had ever seen before. And so they actually discovered several different varieties of bacteria and viruses or diatoms that no one had ever actually even found as an organism. They just found this unique DNA sequence and said, well, there's an organism that has this DNA sequence, and so we found a new organism, and they may or may not have even seen what that organism looks like. So he's actually discovering new species just based on their genomes. The thing I want to talk about that's made news recently is this new company that he's started called synthetic genomes, or synthetic genomics. And what they have done, this was 2010 in Science. They published a paper called The Creation of a Bacterial Cell Controlled by a Chemically Synthesized Genome. What they did is, from scratch, putting nucleotides together, they made an artificial genome. Now, they based it on one of these mycoplasma, one of these bacteria that have got really small genomes. So we've got multiple of these bacteria sequenced, and we kind of know what does it take to make kind of the stripped down basic genome to do cell metabolism. And what they did is took all those sequences and they chemically synthesized them with a machine. They had to use yeast a little bit to kind of get them packaged together properly, but they determined what the DNA sequence was going to be, and they made a completely synthetic genome. And then, so they used as a template the genome of this guy, Mycoplasma mycoides. So they used that as their basis for writing their new one. Okay, so they're just kind of using that as a reference. But they made a completely synthetic one. It's not like they went in and took DNA out of that bacteria. They just used that as a model for writing their own. And then they took a third organism. They took Mycoplasma capicoleum. They removed all the genetic material out of a living bacterial cell. So they took its genome out entirely. And the bacteria could still survive, right? It's still got all of its enzymes, it's still got all of its proteins, it's still doing metabolism. It just for a short period of time can't make any new DNA, can't replicate, can't make RNA. And then they injected their synthetic one that they made completely using biotechnology. And the cell continued to live and then started making RNAs and making proteins all off of this synthetic genome. So they have completely jump-started, I guess, a new organism here, because that thing used to be a Mycoplasma capricoleum, but that cell now doesn't have that genome anymore. It's got the synthetic genome. This is pretty awesome. This idea of synthetic genomes is really the idea is we can now try and custom make organisms to do things we want them to do. There's a lot of interest in this in making materials. So getting bacteria to make you insulin, or making bacteria make you acetone, or chemically interesting products that you can use, or pharmaceuticals. You could also think about having bacteria that will specifically clean up a certain type of toxin, right? If you had a chemical spill, you've got a contaminated lake, you could, in theory, make an organism that goes in and eats up and metabolizes that toxic thing into some non-toxic form. So you could do bioremediation. So there's all kinds of interest in making custom genomes. This got thrown around in the media saying, oh, they've created life. They did not create life. This is not a synthetic life. This is an already existing life, right? That bacteria was already alive. All they did was put in a synthetic genome. So this is pretty interesting stuff. Venter continues to kind of push the limits of what's going on in genome research. Questions about this? Yeah.
So could you hypothetically maybe
Yeah, so what you're talking about is more gene therapy. Is like trying to replace the broken gene that's causing the cancer. That's gene therapy. I mean, there are some ideas of like using synthetic bacteria to treat human diseases, where you could like, you know, just inoculate somebody with a bacteria that's making some product that would treat whatever their illness is. I don't know. The first thing that occurs to me is like lactose intolerance. Like, if you don't have the enzyme for breaking down milk, that's in your gut. Well, what if we made a bacteria that made that enzyme for you, and then stuck that enzyme in your gut, and so then the bacteria would make it for you, and you would alleviate the condition. So yeah, there are medical applications you could think about of doing synthetic genomes. Gene therapy would be, you probably wouldn't want to try and synthetically reproduce the entire human genome. You'd want to just go in and fix whatever mutation was causing your cancer. But it's the same kind of ideas. I mean, gene therapy and custom genomes, it's like, can we change the genome to do something different? Fix what's broken, something like that. When they created this artificial genome, they actually translated English phrases into nucleotide language. They had decided on their own scheme, different than Church's group. But they called it their DNA watermark. And in the paper, they actually like said, we've put a watermark in there and we challenge anybody to go find the watermark and translate it and actually figure out what we said. So there was like this competition going on at the early stages when this paper was released, and people were searching through this artificial genome sequence because they released the entire sequence that they made, and looking for things that didn't match up, that were obviously inserts that they had put in there. And then they were running through like code cracking programs to try and decide what actually was the code that they were using to change an English letter A into what was the nucleotide code for that A. So if you poke around, you can go find, they put like some quotes from literature, and they put all their names of the people that were involved in the project and other interesting things that they coded into the genome. So you could actually make custom organisms. If we're thinking about linking up custom organisms with storage, you could actually make an artificial chromosome that stored some information, put it in a living cell, and get that cell to be living in, I don't know, you could put it under your skin and have the bacteria, with whatever message you put in there underneath your skin, or you could grow it in big vats and store information in living organisms. All right, I'm going to talk about chromosomes now. Up to this point, I've just been showing you DNA, and it's just been, the sugar ribose, the ribose sugar phosphate backbone, and the base pairs pairing together. DNA, unless you've like synthesized it or extracted it from a cell, it almost never exists in that like naked just DNA form. DNA is almost always in living systems, DNA and RNA, almost always surrounded by accessory proteins, holding it into specific structures. So this is a second level, or I guess third level of organization of DNA, right? You have DNA, and the first level of organization is the actual sequence. What sequence are the base pairs in? The second level of organization is, secondary conformation, RNA folding onto itself, DNA twisting into the double helix. Whether it's linear, whether it's circular, that's all like second level. This is now third level organization. How is those actual DNA strands wound around proteins and organized in more three-dimensional structures? So chromosomes is DNA plus all the proteins that go along with it. So DNA is never naked, nor is it really ever just like a floppy noodle, just existing in a linear form. It's always going to be folded or wrapped or spiraled around itself. So we'll start simple first again, and we're going to talk about supercoiling. Supercoiling is what happens in prokaryotic cells. They usually have a circular genome like we've talked about, and the DNA itself is in a helix, so it's twisting around itself. But that twisting helix will often get twirled around itself as well. And this example is becoming more and more outdated because hardly anybody ever has phones with these kind of cords anymore. But if you think about an old school phone with the receiver, and then the cord going up to the base has got a coiled wire, right? It's induced this helical pattern. So this would be the helix, the double helix of the DNA. Supercoiling is when you stretch that out really long and you're, your sister's going underneath you, and this used to actually happen in my house. That cord would start to kind of twist back onto itself, and you'd induce these stresses and strains in it, and it would actually start coiling. The coils would coil upon themselves, coiling upon coiling. That is supercoiling. So where the coils are actually spinning around themselves. It would be like twisting it together on a second level. Okay? Most bacterial genomes are going to be supercoiled. You wouldn't want your entire E. coli doesn't want its entire chromosome just open. There's no room in the diameter of the cell for that. If you actually stretched out the entire E. coli genome and made it actually a big circle, it's going to be bigger than the diameter of the actual bacteria itself. So there's no way you could actually unfold it all at the same time. So you have to densely wrap it up. Now it does this by supercoiling. So twists upon twists. This is actually, this is a tiny little bacterial plasmid under atomic force microscopy. AFM is like using this ridiculously small needle to touch the surface. And so what they've done is coated some of this plasmid DNA on the surface of a glass slide. And they're using this ridiculously fine tipped point to actually like feel the surface of it. So it's giving you a three-dimensional texture of whatever's on the top of that slide. So what you're looking at is a piece of plasmid DNA on the top of a slide. And what they did is just induced supercoiling by changing the medium. So this is an unraveled, very small plasmid. And then here you can see it starting to supercoil on itself, even tighter and tighter and tighter. This actually is 2 strands then of DNA really, really tightly wound around itself. Okay? So that's the first level of organization that prokaryotes do. They supercoil. Now, that's still not enough to get it small enough to even exist within the cell, even if you supercoiled it all. So what ends up happening is you take those supercoiled regions and you start looping them back on each other. Okay? So I've got the helix that's supercoiled on itself, and then I start looping that back and forth, folding that on itself. I'm just really trying to shrink down this to the smallest diameter I can. So this is another AFM, atomic force microscopy image, of the entire bacterial, E. coli bacterial genome. Okay, so instead of just being a plasmid now, this is the entire genome. And each one of these strands that you're seeing out here, that's actually supercoiled DNA. Okay, so that's 2 strands supercoiled on top of themselves. And then they're looping all, this is, if I could actually unravel this, you can kind of see it maybe a little bit here. So that is supercoiled. And it's supercoiled stuff that's actually looping back on itself as well. And there's a bunch of proteins in here that are all holding these loops together. Right? So it's almost like, I don't know, genomic knitting or something like that. You've twisted your whatever your fabric is, or whatever your wool or hair or fiber, or whatever it is. You've twisted it into yarn, and then you're stitching the yarn and moving it back and forth and just really compacting it down. To do that, you have to keep it in position. And we don't do that by knotting it like if you're knitting. We wrap it around proteins in there. So it's kind of like wrapping it around a spool. So this is the extent of organization that prokaryotes do. There's no nucleus. It's a relatively small genome. So supercoiling and looping is going to get you small enough to keep it in your cell. And then the cell will selectively unravel the sections that it needs. So if it needs to do a certain type of metabolism and needs to express a certain protein, well, it unravels that little portion of the genome, makes an mRNA, turns it into protein. When it doesn't need that anymore, supercoils it down, loops it back in, and it can open up another thing. So it's kind of like an organizational file drawer system. If you don't need that protein, you just save the DNA away in a really condensed form, and you open up whatever files you actually need at the time. Okay, so that's bacterial, prokaryotic. Next time we will talk about eukaryotes. And as you expect, we're going to get bigger and bigger genomes, and we're going to have higher and higher levels of organization and condensation of packing this material together.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.